Hi, it's Mr. Rikio. So today we're going to talk about C1.1, enzymes and metabolism. So this topic covers how enzymes work and what is the relationship with the metabolism. I'm going to start with the center level, and then we're going to cover the additional high level for this topic. So first, defining metabolism. The metabolism is a complex network of chemical reactions. In other words, all the chemical reactions that exist in your body, all the biochemical pathways, all the processes that generate energy, that consume energy in the human body or in a cell or in a tissue. All right. Here we can see some examples. So the mitochondria, the chloroplast, they're perfect examples of metabolic areas where a lot of metabolic processes happen in our cells. So metabolism is this very broad concept in biology. Okay, it encompasses all the biochemical reactions. And metabolism typically involves anabolic reactions and catabolic reactions. So in anabolic reactions, you're building more complex structures. You're combining monomers of molecules, for example, saccharides, for example, carbohydrates, into polysaccharides. And as we can see in this example, or you can make peptide bonds to make polypeptides. So you're building something more complex. You're combining molecules to build a polymer. All right. Again, this is typically done and promoted by enzymes. Nothing happens by chance in the cells. Okay, most of the process they don't happen by chance. In other words, the opposite of anabolism, we have catabolism, when you're catabolizing a structure. So catabolism involves the breakdown, the digestion, typically through hydrolysis reactions, the catabolism of structures. So if you have an enzyme that comes here in this oligosaccharide, and if you break down these covalent bonds, you're releasing the monomers. So you're breaking down from polymers to monomers. All right. So this is typical hydrolysis and involves catabolic reactions. It's a very simple comparison. One is the opposite of the other. And if we check some examples, for example, anabolic reactions, protein synthesis, if you're growing, if you're becoming stronger, you're synthesizing a lot of proteins in your ribosomes. So this is anabolic. Glycogen formation, if you're storing, if you're accumulating in the muscles or in the liver, this is also anabolic. Or photosynthesis, the most important process of life on Earth, or how plants make organic compounds using the sunlight as a source of energy. All right. In opposite to anabolic, we have catabolic. So, for example, hydrolysis of macromolecules by digestion, the digestive enzymes in our digestion is a perfect example of catabolism. All right. So we are using our enzymes to break down all the starches, proteins, and lipids that we're consuming. Or also the oxidation of substrates in the mitochondria, for example, glycolysis, the Krebs cycle, until the electron transport chain. So these are examples of catabolic reactions. You're digesting, you're breaking down more complex blocks into smaller chunks, into smaller monomers. All right. And as I said, these reactions, they are promoted by enzymes. So the enzyme is a protein. We basically have two types of proteins in biology. Okay, we have the structural proteins and the enzymatic proteins. So the structure is like your body, okay, your tissues, your structure, like collagen, for example. And the enzymatic proteins, they're enzymes. They promote reactions. They promote processes that are vital for life on Earth. Here we have an image, a 3D structure of pepsin, okay, this digestive enzyme. So we see all the amino acids that build this complex 3D structure. And we can also see the active site in this region. So the active site is where you're actually going to have the reaction, what actually is going to trigger the process that you're looking for in an enzyme. Typically, enzymes, they are globular, so they form like a globe, a circular 3D structure. They have an active site that is crucial for the activity. And they have just a few amino acids interacting in this active site. So typically, you can change the amino acids in the active site. If you change the amino acids in the active site, you lose the function of the protein. And the interaction between all these dozens or hundreds of amino acids is going to dictate how this final structure will be shaped. And this shape, this form, is related to the function. If you change the form, you change the function. All right? One model of how enzymes work is what we call the lock and key model. It's a simplified model that describes how the substrates they fit, they lock and key in the active sites of enzymes. So we can see here, for example, let's suppose this is lactose, okay, the sugar present in dairy products, milk, cheese, etc. And this is lactase in your intestine. So the lactase is going to interact with the lactose. They're going to fit on each other. Okay, there will be a fit. All right. And then the lactase is going to promote with the active site the actual reaction. That is a hydrolysis, the chemical hydrolysis of the lactose. Okay, when you break down the bonds, you have two products. All right, and then you're going to release these two monomers. So you make a catabolic reaction. You make a hydrolysis and a catabolic reaction, breaking down one D saccharide into two monosaccharides, that in this case would be glucose and galactose. All right, this is the model. This is one of the most common models of enzymes, of how enzymes work. Okay, the lock and key model and how they fit on one another. All right. So again, making it like a more realistic approach, a 3D structure, we have the binding site, the active site, the substrate locks, that is interaction, and then the hydrolysis of lactose, of lactase into the two monomers, into two monosaccharides. All right. When we look at the thermodynamics of enzymes and the rate of reactions and the catalysis of reactions, all right, why enzymes are so important? Because they reduce the activation energy, the energy necessary for the reaction to occur, and they make it faster. In other words, they make it faster and they make it more efficient and with less energy. This is why enzymes are crucial for all life processes. And in fact, several processes that happen in biology, they wouldn't happen without an enzyme. All right, some process might happen without an enzyme, but most process they wouldn't even happen without an enzymatic action promoting the reaction. All right, so here you can see the two curves, one without enzyme and one with enzyme. So we can see how efficient it is when we compare in terms of level of energy and the speed of reaction. Now talking about the factors that influence the enzymatic activity, the rate of reaction. First one is temperature. Typically, more thermal energy, more temperature, more reactions are going to have. Because more enzymes, more active sites are going to lock in the substrate. So more active sites, more substrate, more reactions. If you have more movement between the molecules, more reactions you're going to have until you reach an optimal temperature or best temperature. This is where you find the maximum rate of this reaction. Okay, typically in organisms and tissues and cells, this temperature would be somehow around 37, 38, 40. It depends from protein to protein, but a lot of proteins, they are around this range, including us. So if you continue to heat these proteins, they will start to change their form. The amino acids, they will be so stressed that they will start to change the form. If you change the form, you change the function. So if you change the function, in fact, you lose the function. So we see how the enzyme activity drops quickly. And in this case, the cell is dying because you're not having the enzymes for the reaction. All right. Second one, the effect of pH. So different proteins might have different optimal pHs, and this is totally normal. Some of them work best on acidic pHs, some of them work best on alkaline pHs, some of them work best in neutral pH. It depends from protein to protein. Typically, most proteins, they work well in the neutral pH, so around 7, 6 to 8, around neutral 7, they work best. Here, for example, we see that this protein blue is working greatly on pH 2, very acidic. Probably this could be an enzyme in our stomach, okay? So thriving the stomach acid digesting proteins, for example. And in the other hand, we have a pH of 8, probably. So this could be an enzyme working in the intestine, the small intestine, digesting other compounds from our nutrition, like starches or lipids. Okay, so both have different optimal pH values. All right. Next, we have the concentration of substrate. So the more fuel you give for the reaction, more products you're going to have. So the more substrate you give, more reaction until all the active sites of all enzymes are busy. They're occupied by the substrate. So you can continue to grow forever. You have a limit, you have the maximum rate, and then you can stabilize, and then you continue on this reaction, on the maximum speed. Okay, because everybody's busy, you can do more, right? Unless if you add more enzymes, then you're changing all the environment. Now talking about the additional higher level. If you're center level, that's it for you. You cover all the center level concepts for this topic. But if you're higher level, let's check the additional higher level. So we start talking about the extracellular and intracellular enzymes. So there are enzymes that are built to stay inside the cell, and there are enzymes that are built to be secreted, to go outside, do their job outside of the cells. So we're going to talk about them, all right? So the first one are the extracellular enzymes. So they're produced inside, of course, they're transcribed and they're translated in the cytosol, the cytoplasm, okay? But they're let's say, fused with vesicles, internal vesicles, to be released outside of the membrane, of the cell membrane. So they're put outside so then they can do their job. Like here, you have an exocrine gland, and then these proteins are being put outside. The best example are the digestive enzymes. Okay, in your intestine, they're released outside to work outside of the cell. Or your saliva, the amylase in the saliva, it's also another example. All right. If you see the cell, so again, it is translating inside, put in the vesicles, and then through exocytosis, it secretes to the outside. All right. The best example, digestive enzymes, as we discussed. Next one, we have the intracellular enzymes. So they're, of course, they're produced inside, but they never leave. They stay inside the cell and they do their job. They do their metabolism inside the cell. Very famous examples are, for example, the enzymes in glycolysis, Krebs cycle, or all the ones in chloroplast or mitochondria. They are involved in pathways, biochemical pathways, complex biochemical pathways inside the cell, inside the cytoplasm, or inside an organelle, such as the mitochondrium. All right. So this is the first distinction of additional higher level. Next and final concept, we need to talk about enzyme inhibition, higher level. All right. So every enzyme needs to be regulated. Otherwise, you're going to have a mess in terms of reaction. As everything in our lives, everything that we produce needs to be regulated. For example, in a factory, enzymes need regulation as well. So then they can work properly, and then you can have this refinement and fine balance of biochemical pathways. So the first one is the competitive inhibition. The competitive inhibition is when the enzyme with an active site digests a substrate, but the product of this reaction that is similar to part of the substrate fits and competes with the active site with the substrate. So this product, this competitive inhibitor, is similar to the substrate and competes for the active site. So the more products you have, the slower your reaction is. So as you're producing a lot, you have a lot, you don't need to continue to produce so much. So you reduce your efficiency. So it's a way to balance how much products you have. The more products, the less reaction. The less products, the more reaction you have. So it's a fine balance that cells achieve through competitive inhibition. All right. In contrast with competitive inhibition, we have non-competitive inhibition. I make a joke that they're not even competing because it's so effective that you don't even need to compete. You just win. So what is a non-competitive inhibitor? It's a chemical compound. Okay, it can be a drug or it can be a chemical compound that is going to bind to the enzyme, another region of the enzyme, and all other amino acids. It's going to bind to the enzyme and completely change the 3D conformation of this enzyme, consequentially changing the active site of this enzyme. And as a consequence, the active site no longer fits, no longer binds to the substrate, and you stop the reaction. So it's more effective in terms of rate of reaction. Okay? When we see a graph, and this is the final idea, when we see a graph of rate of reaction versus substrate concentration, this is what we see. So we have the blue line uninhibited. Okay, we follow the exponential curve. There is a growth. And as all the active sites are busy, you stabilize on this number of Vmax. If you have a competitive inhibitor, you have a slowdown, you have a reduction in efficiency. So you reduce the Vmax, you reduce efficiency, but eventually you reach the same maximum rate. But If you have a non-competitive inhibitor, we have a difference here. Okay, we have a large difference in terms of efficiency. So the reduction, the control of the reaction is way stronger and way more efficient. So we see that the speed is very slow and the efficiency of the reaction drops quickly. So that's it for enzymes higher level. I hope it was helpful.